So as always, I have the learning objectives for this lecture. It does look like a lot of terms, so a lot of it is some memorization of terms, but also understanding how the terms relate to each other. But all of these are very important terms for those of you who are interested in disease, whether it's studying it from a clinical perspective or whether it's studying from a research or public health perspective. And if you have any questions about the content in this material, I'll be available during office hours and also happy to answer questions during class or lab if anything that I can clarify. The first thing we'll start with is terms we can use to describe microbial associations. So many of these terms, if you've taken a class in ecology before, you'll recognize them because we can use them to describe other types of associations between organisms. So we'll start out with what we call the symbiotic, or if you've talked about this in an ecology class, symbiosis, which are when organisms are living in close relationships. So in general, we're looking at nutritional relationships. So for example, we talked in the case of the root nodule, where the microbes were in symbiosis with the plant on which it was forming the nodule. Now, these can be both negative or positive for the organisms involved. So in the case of the root nodule, that would be an example of mutualism. So in other words, both organisms are benefiting. But in the case of parasitism, imagine a parasite like a tapeworm living in a human. The parasite is dependent on the organism and is benefiting, but the host is being harmed. In the middle, we have commensalism. So you can imagine an organism growing on your skin. It's good for that organism, it has a place to live, maybe getting some nutrients, but the other member is not harmed. So you're not harmed by the microbe living on your skin. Now some of these associations can be a little tricky to classify. So for example, even though the case of the microbe living on your skin seems like perhaps an obvious commensalism, you may be obtaining some benefits as well. For example, the presence of harmless bacteria on your skin, such as harmless Staphylococcus, can prevent more harmful species like Staphylococcus aureus from growing. But what I want you to know about these terms is thinking about the benefits. So think about a plus minus and plus plus. So for example, mutualism would be plus plus for the organisms involved. Commensalism would be plus for one and a neutral for the other. And in parasitism, one is benefiting as a plus, but the other is being harmed, so it would be a plus minus. You can also have what we call non-symbiotic relationships between two free-living organisms. About the symbiotic relationships, you're thinking about close relationships required by one or both members. So in other words, in parasitism, the tapeworm needs to be living in the organism. Here, these relationships may not be required for survival between the free-living organisms, but they are still examples of associations and interactions between the organisms. So for example, in synergism, members can cooperate and share nutrients. You may have some cooperation between microbes, different species of them, in a biofilm. In antagonism, some members can be inhibited or destroyed by others. A good example of this could be some microbes make natural antibiotics. So for example, some molds, like penicilliums, do. By secreting these antibiotics out into the media around them, they can inhibit the growth of other species. So just to talk about this a little bit more, as I mentioned, you can have what we call normal flora, which are the microbes living on or in surfaces of the body. Now, of course, most of your body is going to be free of microbes, so your tissue should in general be sterile. If you have cases where that's not happening, so for example, if you have microbes in the blood, that can be the indication of a serious infection. And soon in this lecture, we're going to give some examples of what normal flora would be. And then again, we talked about this idea of parasitic. So one organism is using another organism. So pathogens can be considered parasitic in that, for example, a pathogen invades your body. It may invade sterile tissues inside of your body. It can multiply, it can use nutrients from your body, and it can lead to infection. And what happens in the course of what we would consider an infection is one of the things that we're going to consider today. Back to the idea of normal flora. So while most of our body does not naturally have microbes, nor should it, because that would be the example of an infection, we do have many surfaces on our bodies that have normal flora. So there's whole microbial communities that are living naturally. For example, our skin has quite a community of microbes. Our upper respiratory tract, and we've talked in class before about our gastrointestinal tract. We talked about this a little bit as we talked about antibiotics and how taking antibiotics can disrupt the normal flora of your gastrointestinal tract. So we're going to look on the upcoming slide at a few examples of what this normal flora would be like. So for this end, I decided to focus on one example, which would be the normal flora of the gastrointestinal tract. So this includes parts of the body that have microbes in the oral cavity and the upper gastrointestinal tract, as well as microbes in the large intestine. So just to give you a few examples, there's many examples of Streptococcus species that are naturally found as normal flora of the mouth. Now, we've talked in class about some forms of Streptococcus being pathogenic, but there are others who are naturally found there, and some can be involved as well in dental caries or dental cavities. So Streptococcus mutans is one example. In the large intestine, you can imagine that conditions there would be low in oxygen, and so that would favor the growth of obligate anaerobes, going back to the terms we learned earlier in the class. So Bacteroides is a very common resident of the large intestine, but there's a range of forms that can be found, including some species of Clostridium. So if you're interested in learning more, the book has lots of info about the residence of other parts of your body. So for example, the upper respiratory tract and the microbes present on your skin. So each of these is going to be adapted to the conditions. So in this case, we talked about obligate anaerobes, but you can imagine the conditions on the skin are going to be dry, and also they happen to be relatively salty. So the microbes, such as types of Staphylococci, such as S. epidermidis, which is a Staphylococcus that's harmless, is going to be adapted to these conditions. So now that we've talked about normal flora, let's move on to some information about pathogenic organisms, so the development of an infection. So the first thing that we'll talk about is true versus opportunistic pathogens. So a true pathogen is going to be capable of causing disease in a healthy person with normal immune defenses. So this could be bacteria, viruses, fungi, parasites. Think about things like influenza. So many of us, we have normal immune defenses, we're healthy, but we can still become sick. In contrast, an opportunistic pathogen, so the term comes from the idea it has an opportunity. So in other words, it's not going to generally cause infection unless there's an opportunity. So someone who has compromised defenses, so this could be someone who's immunocompromised or compromisation of the immune system due to taking certain types of drugs to suppress the immune system for certain cases, or maybe they have another type of infection, so maybe they've developed AIDS and so they can develop opportunistic infections, would be good examples. Or they might be introduced into a part of the body that's not natural, so maybe due to some sort of hospital or medical procedure, you can now introduce pathogens that normally wouldn't be in a certain part of the body, but they're able to grow there. So in other words, opportunistic pathogens, if we were working with them in the laboratory, we normally wouldn't expect that they could cause disease, but under certain conditions, they can. Now, another term that's important to think about is the idea of virulence. So you've talked about this a little in class, use that term maybe virulent, and it really is referring to the severity of the disease. So you may have different strains or forms of a particular species of bacteria, and some of them can cause more severe disease. And this is due to the presence of things called virulence factors. So virulence factors are characteristics or structures that contribute to the ability of a microbe to cause disease. I'll give you a few examples. So we've talked in class about different types of toxins, which are proteins that bacteria can secrete or move outside of the cell that can then harm the body. Other virulence factors could include a capsule. So we've talked about the capsule as a layer around the bacteria earlier in the class. that can help the bacteria evade the immune system. And we're going to be talking about the immune system in the coming weeks. But these are both examples of virulence factor, characteristics or structures that help the bacteria to cause disease. Now, these terms, as well as more that we're going to be talking about today, may be helpful to you as you prepare your oral presentations. One of the things you may be interested in looking at are what are some of the virulence factors that are produced by your pathogen that you've selected for your presentation. Back to this idea of an opportunistic infection, there are certain factors that can make someone more predisposed to have an infection, like a bacterial infection. I'd like you to know at least 3 of these examples. So these could include old age or being very young. So often you'll hear about for certain diseases, like certain types of the flu, the elderly are more predisposed to infection, or young children as well. There may be genetic differences, so some people have genetic differences in immunity, or maybe they aren't able to make certain types of B or T cells that we're going to talk about in upcoming lecture. It can be more transient or temporary conditions, so after surgery you may be more predisposed, and individuals who've just had an organ transplant are generally on drugs that suppress the immune system to prevent their body from rejecting the organ. So this can increase greatly susceptibility to infection. Other diseases as well, so for example, individuals with diabetes are more prone to certain types of infections. As I mentioned before, immunosuppressive drugs for different types of conditions will increase the ability of getting infection, especially opportunistic infection. Physical and mental stress, and the presence of other infections. So a few examples of opportunistic infections were different fungal diseases, as well as cancers that were developed by individuals who were infected with the HIV virus and then had progressed to AIDS. So some of these, and this has actually helped lead to figuring out and studying the early origins of the HIV virus, because those were infections, they're opportunistic infections that were normally not seen in young individuals. So when there was a cluster of individuals with some of these unusual diseases, including unusual fungal diseases and Kaposi sarcoma, clinicians noted this, and this led to additional research by scientists to identify HIV. Looked at who might get an infection, and said it can vary. We are susceptible to infections at many points, whether we're healthy or immunocompromised. Let's look at how an infection develops in the body. So I think this graphic from the book does a nice job of kind of showing the process over time. So first, the microbe must get into the body, and this can happen through a variety of what we call portals of entry. So it can be through a cut, it could be through a mucous membrane, so through the eyes or your mouth. You might eat a food that has a harmful food pathogen, so it enters through the gastrointestinal tract. So these are all different types, and it's going to depend on the microbe. And then the microbe needs to attach to your host cells. And we've learned, I just want to kind of tie this back to some of the things we've talked about before in class, this process of sticking or adhesion can be helped by structures like fimbriae. So remember those fimbriae were those kind of like arms sticking off of the bacteria cell that can help with adhesion, and capsule, so that sticky layer of polysaccharides around the bacteria. Those can each help with this process of adhesion. Next, the bacteria are going to need to invade. Okay, so they're going to need to make pathways into cells, into your body, and into normally sterile areas. So, as I mentioned, we're going to be talking after Thanksgiving about the immune system. And bacteria are going to need to block clearance. So by that, it means your body is going to be trying to get rid of these invaders. So it's like a war between the bacteria and your body. And if the bacteria wins out, you get a serious infection. So the bacteria needs to do things to block being cleared or being removed. And instead, it wants to multiply and grow and spread. And this is where those virulence factors come into play. So the bacteria uses virulence factors to invade your tissues, to colonize, and as I mentioned, it can include toxins that are made by the bacteria. If the bacteria can invade your immune system and actively invade, it will be able to grow and spread by multiplying, by binary fission, and then it'll attack tissues. And when it's causing damage to your body, this would be when we would consider the process of disease. And at the end of the lecture, we'll talk about exit and spreading to others. So the microbes then can leave the host and spread to the next person or other organism that will be infected. So the things that we're talking about now, certainly things like mucous membrane would be specific to a human or an animal, but plants, for example, can get infected as well, and there's going to be some commonalities. So while there won't be a gastrointestinal tract, microbe can enter the host, maybe through a cut on the plant, and the microbe is going to need to use these same type of processes to cause disease and damage to the plant. Look at this information as well if we're looking at the symptoms that a person is experiencing after infection. So it's going to be initially exposed to a microbe, and then usually there's a period. And you're probably familiar with this just from thinking about diseases called the incubation period. So, you imagine your roommate is sick, and then it takes a few days for you to become sick. You may have been exposed to that microbe at day 0, but depending on the disease, there's different lengths of incubation period. So some of them can even have incubation period of a month. Things like the common cold or the flu are going to have much shorter incubation periods. And then you're going to have an increase in the intensity of symptoms, including here you have the height of the infection, the bacteria actively invading your body, and eventually symptoms, unless it's a very serious infection, symptoms should resolve and the intensity of symptoms should decrease. But again, remember that the microbes are going to be exiting, leaving your body, and spreading to another organism. Maybe some of your friends in the class, they're going to have their own incubation period and period of symptoms. So some more terms here, and of course, again, this lecture is a lot of terms, which is one of the reasons why I've made this a video lecture as well. I think it's a good selection for that one, because you can go back and review some of these terms. So again, though, make sure that you review some of these before your oral presentation, because I think that will help you a lot using the correct terminology when you're describing your microbe. So another important term to think about is the infectious dose. So the infectious dose is the minimum number of microbes that would be required for an infection to proceed. So in other words, let's say you're on a cruise ship and you've received some food that's infected with the Norwalk virus. So it causes severe vomiting, and it's something that you really don't want to get, but it's very infectious. And what happens is you only need a small infectious dose. So you only need a very small number of virus particles to become sick. That's not true for all diseases. In some cases, you need a lot more microbial cells or virus particles to become sick. And so if you don't have an infectious dose, that will not result in infection. But my example from the Norwalk virus is that you can get sick in many cases from only a few microbes, so only a few bacteria cells or only a few virus particles, because remember, they can replicate in your body. And so even if you are able to kill most of the microbes in a food or something else, that may not be enough to prevent infection. Some additional terms are the idea between signs and symptoms. And this is something you may not have thought about, the distinction, but there is a clear difference between what we consider signs and symptoms in clinical description. So a sign would be, the definition is objective evidence of disease as noted by an observer. So think of it as things that if you're an outside person, like let's say you're the doctor, you can objectively look at a person or do a test to determine if they have this property. So for example, fever you can measure to see if someone has a fever with a thermometer. If the temperature is above the threshold for a fever, then you can mark that off as a sign of disease. Or a rash, so you can observe that a person has a rash. Or you can do molecular tests to look at changes in white blood cells in the blood, or levels of antibodies, etc. In contrast, a symptom would be subjective. So in other words, the patient is sensing it. It's something that you're going to need the patient to report to you. It would be things like headache, fatigue, itching, nausea, aches, pains, or chills. And then when you have a patient describing a set of signs and symptoms, you can consider this a disease syndrome. So a syndrome would be defined as a set of signs and symptoms that is characteristic of a disease. So in other words, if you see this syndrome, all these signs and symptoms together, this will help the clinician in diagnosing the disease. Now, when we think about an infection in the body, we can use a variety of terms to describe it. We can consider it localized or systemic, and we can consider it acute or chronic. So let's see what these mean, and then we'll look at a picture on the next slide. A localized infection would be when microbes enter the body and remain confined to a specific tissue. So a good example might be an infection on a specific spot in the skin. The microbes are remaining confined to that area, like a boil, which can be caused by Staphylococcus aureus, basically a specific spot on the skin that's infected. In a systemic infection, though, the infection is spreading to several sites. It's spreading throughout the body, and tissues and fluids. So, for example, in the bloodstream, you might see signs of the microbe. So an acute infection versus a chronic infection. So an acute infection would come on rapidly, and you have a severe peak of effects, but it's short-lived. So let's say you have a really nasty cold. It comes on, you have a couple of days where you're very sick, but it's short-lived. So a couple weeks later, you've totally forgotten you've had the cold. Whereas a chronic infection is going to progress and persist over a long period of time. And so you can also talk about acute and chronic in terms of other types of diseases. So for example, even though it's not an example of a bacterial infection, a disease that would be chronic would be something like diabetes. So it's something that people will have for a long period of time and manage it, as opposed to something that is short-lived. Some images that summarize a little bit about what we mentioned on the last slide. And there's some other additional terms that you don't just need to worry about the ones on the last slide. But here you can see, for example, they're mentioning you can have mixed infections with different types of microbes. Here you see the different shapes we learned about earlier in the class. But here's what I'm talking about the localized versus systemic infection. So here you can have a boil, so a localized place on the skin where you have infection. And then here would be in a systemic infection, you have influenza, which is spreading throughout the body. And so, of course, there might be cases where a localized infection could become systemic. So imagine you have a wound and you have microbes in the wound and they can spread throughout the body. All right, so we've learned lots of terms so far. So we do have a few more, but we're moving on to the last component here where we're thinking more about the spread of microbes. So in other words, you have that cold that your roommate gave you. Now you're going to spread it to others in your classes. So how does this happen? So there's a lot of different ways where you can spread microbes, and we're going to learn some terms to describe them. And again, this can help you for your oral presentations because you can describe how your disease spreads. So the first term to think about is a reservoir. So a reservoir is defined as the primary habitat of a pathogen in the natural world. So what does that mean more practically? Well, soil might be a reservoir for some types of bacteria. So that's where you would find them, and maybe contact with certain types of soil could get you sick. You may have humans or animals as a reservoir. So some diseases are what we call zoonotic. You may have learned about those in other classes, or you may be interested in them, especially if you have interest in veterinary and medical careers. So there's some diseases where animals can carry them and then spread it to humans. We might have water, might have contaminated drinking water that spreads the disease. Plants may be a reservoir, so there's many possible reservoirs in the natural world. And then the source is just as the name maybe suggests, an individual or an object from which an infection is acquired. So if you got sick from your roommate could have been the source of the disease. Speaking of sources, you can have individuals who are what we call carriers. Carriers may not be sick, but they can spread a disease. And this can happen with a range of things. For example, you can have people who are strep throat carriers, and they may not be sick, but they are carrying that pathogen and can spread it to other people. They may or may not have experienced disease. So maybe they had the disease, and they're better now, but they can still spread it. And we saw some examples of this in the news with Ebola. One of the concerns was that even after people were, had healed from the disease, if they made it through, the concern is, could they spread the disease to others? Could they still carry some infectious particles? And a very famous example would be the case of Typhoid Mary. So she is someone who served as a cook in the New York area, and she had spread the disease typhoid to many people because she was a carrier. So she wasn't actively sick at the time. And actually, it's a very sad story because she had a hard time believing that she was spreading the disease because, obviously that would be very traumatic to find out. And at some points, she was actually institutionalized against her will because she was still actively spreading this disease. And so you can have some different types. The main thing I'm concerned about for the test, the final exam, is going to be the definition of a carrier. But I wanted to give you two pieces of information for your own sake. So you can have what, if you hear in the medical term, someone's described as an asymptomatic carrier. They don't have symptoms, that's asymptomatic, but they can still spread the pathogen. And that's what I was sort of talking about above, someone who had strep throat bacteria that wasn't actively sick, but was spreading it to others. You can also have a passive carrier. And so an example of this would be someone who's a healthcare provider. Maybe they have pathogens that they're carrying on their skin. So you might have maybe colonization of your skin with some MRSA, so resistant Staph aureus, and they can transfer them to other patients. And that's why hand washing is so important, right? Because we want to make sure we're not transferring pathogens to other patients. It's a very important thing. Of a way you can spread a disease is through a vector. So you might have heard that term vector if you've taken physics so far, but we're talking about vectors in the disease sense. And the definition from your book is that it's a live animal, so other than a human, that transmits an infectious agent from one host to another. So to make sense of this, let's look at two practical examples. And I do want you to know these two terms, which are biological and mechanical vector. So a biological vector is identified as being actively participating in a pathogen life cycle. So what does this mean? So let's look at our picture of our mosquito on the side. The mosquito is a biological vector. So the mosquito, let's say, is transmitting something like West Nile virus, or it can transmit something like malaria. So in the case of malaria, when, let's say, it picks up the pathogen, it becomes infected. So it's actively participating because it becomes infected by the organism. And then when it bites the next host, it can pass on some of that organism. So you can imagine that this can help in terms of replicating the microbe in the biological vector. And as we mentioned in some previous classes, mosquitoes are a growing problem. There's a lot of concern about spread of viruses in particular. And there's other insects as well that can be a concern. So in the news just this week, there was some reports of Chagas disease spreading from a bug that's found in Texas, and people were getting sick. So there's a growing concern about the spread of biological vectors. So one of the things is that as areas become warmer, so you can imagine in the winter, normally it'd be cold, you'd kill off some of the insects, and then they'd have to basically re-move into the area or restart the life cycle the next year. But if it's warm enough that these insects can persist during the winter, you can get growing pools of insects that are infected with these diseases. The contrast would be a mechanical vector, so that's shown here. We have a fly. So imagine that you have some trash, and maybe the trash is contaminated with salmonella bacteria. Then the fly lands on the trash, and it picks up on its feet. I guess at the end of its legs. That's probably not a very entomology appropriate term. But so it picks up at the end of its legs some of the salmonella microbes. And then it can move to another area. Maybe it lands on your food, and it spread the agent. Now, it's not necessary to the life cycle, so the fly itself doesn't become infected with salmonella, but it can transport it, and it can help spread things as well. So it's important to consider both biological and mechanical vectors. As I said, you have a lot of cases where you can have insects that can be the source of spreading of viruses in particular, but as parasites as well, and some, as we saw in the example of the mechanical vector, you can also have bacteria spread that way. To the last component, last couple of slides, which relates to transmission of diseases. So we just gave a few examples of vectors, but let's look a little bit more broadly at the ways you can transmit a disease from one person to another or from a source to another person. So we have some communicable diseases, and they can be divided into direct and indirect mechanisms of spread. So direct mechanisms would be direct contact, such as kissing, a way to spread Epstein-Barr virus, which causes mono, if anyone has had mono. So droplets, so colds or chickenpox, so you have droplets that maybe someone is sneezing, coughing, can be spread to a person in close contact. Vertical transmission, so that would be spread from a mother to the child. So you can have vertical transmission of HIV. So that's one of the reasons we'll check for if a mother does have HIV. There's things you can do to reduce the amount of vertical transmission significantly. It's an important intervention. And we just talked about the biological vectors, right? So the biological vectors can spread West Nile virus or malaria to someone. And so there's a lot of things we can do to prevent that. You know, if you're in an area where you have a lot of these biological vectors, you might have mosquito netting, or you might use bug sprays as well. Now on the other side, we have indirect mechanisms. So fomites is a term we talked about in one of the first labs. We did the fun with fomites where we swab different surfaces like doorknobs. So fomites can pick up microbes, and as long as they can persist, even if they're not actively dividing, if someone touches the knob, you can pick up microbes like Staphylococcus aureus. You can also have contamination of food, water, biological products, so this foodborne infections like salmonella, or the pathogenic forms of E. coli can be spread in meats. Water can be contaminated. We talked about the spread plating and looking at levels in milk or water. So there's a lot of different types of pathogens that can be spread in this way. And not just bacteria, but also parasites and viruses. Then you can also have aerosols, okay? So you basically have these small drops. They're specific sizes. They can be suspended in the air, okay, and they can spread, and let's say you breathe them in, they can contact mucous membranes. And so things like tuberculosis, the influenza virus, and hantavirus, which is a disease that's common in the Southwest that's spread by mice, are all diseases that can be spread by aerosols. So aerosols could be produced, let's say you're working in the lab, and then you spill a tube of tuberculosis bacteria. You can have these droplets in the air form an aerosol, and you can breathe them in. So diseases that can be spread by aerosol are a big concern for people working in labs with different pathogens. You might have to use a special biosafety cabinet which prevents you from being exposed to the aerosols to prevent yourself from getting sick. So diseases that are caused by aerosols are a particular concern, not just for people out in the environment, but also for lab workers who are working with diseases that can be spread in this way. Slide is just a summary of the things I was mentioning on the last one. It provides another summary if it's a little harder to see the information on the picture of the direct, which is through contact with a living organism, or the indirect mechanisms, which is through a separate vehicle. So you can see the indirect, we have aerosols, so in the air, food or water, and fomites. So those are going to be through a separate vehicle. So the droplets we put in the direct because it's from very close proximity from a sneeze. You have the droplets going very close to the next person. And this is a very final slide, kind of showing this. It's a little gross perhaps, but it's some pretty amazing photography looking at the process of a sneeze. And you can see all the droplets. So all those droplets are going maybe to a nearby person. And in some cases you can have some particles that are of a size that can persist in the air and spread by aerosol. But remember that not all diseases spread by aerosol. So different diseases are going to have different modes of transmission. So that's one of the things you can look at for your oral presentation. So how is your disease spread? Is it spread through indirect? Is it spread through direct? Is it spread through multiple mechanisms? What are some of the reservoirs of the disease? So do they get it from other people, and or do they get it from animals, and or do they get it from contaminated food or water? So you can look at all of these different components. So that concludes our video lecture for today. So what I'm going to do is post a separate lecture worksheet that's totally optional, but it gives you a chance to practice using some of the terms that we did in our lecture today. I'll give you plenty of time in the upcoming weeks if you have questions about any of these terms. And as I said, I hope we'll have a chance for you to practice these as you put together your oral presentation. And we'll be using some of these terms as we talk about some diseases in the lectures ahead. We'll be moving on to next week is thinking about viruses and the way they replicate. And then we're going to be concluding the class with a couple of different topics. So we're going to talk about the immune system. We'll talk a little bit about vaccines. And then we'll talk about, finally, at the end of class, epidemiology. Epidemiology looks at disease not in just an individual organism, but in a population. So for those of you interested in public health and the spread of disease in populations or the prevention of disease in populations, you should enjoy that last unit. So just a couple of more weeks left to consider some of these topics. And remember, the information on this video lecture is something that you will need to review for the final exam.